Okay, I have exactly 12:30 Central Time, so we should get started now. So welcome to today's webinar, Sudden Death Syndrome, Risk and Management. My name is Laura Isles. I'm one of the co-directors of the North Central IPM Center. We're one of 4 regional IPM centers supported by USDA NIFA Crop Protection and Pest Management Regional Coordination Program. All of the centers have a mission to coordinate IPM across our regions by sharing and promoting IPM research, extension, and teaching activities. And one of the ways we do this is through this Pests in Progress webinar series. So as Daren's speaking, if you have questions, please type them into the Q&A part at the bottom of your Zoom screen, and he will answer the questions at the end of the presentation. So our presenter today is Dr. Daren Mueller from Iowa State University, and I'll let you take it away, Daren.
Thanks, Laura. And so, yeah, my name is Daren Mueller. I'm at Iowa State University, and I'm an extension plant pathologist. And today's talk is going to be covering sudden death syndrome, which is a disease on soybeans. And we're going to be going over the management of it, but then ultimately what we want to get to is assessing the risk for sudden death syndrome. And then how does that, knowing that risk, how does that affect management decisions? All right, so to get started, I'm just going to say thanks to all of the colleagues. There's a long list of people that have either contributed data or ideas. The grant that I'm going to be talking about is one that was funded by USDA NIFA, and my colleagues that were on it are Nathan Klacheski, who was at University of Illinois, and Marty Chilvers at Michigan State University. And then we have grad students, Edward Ernett here at Iowa State, and then Ryan Hamilton at Michigan State are the grad students that are working on some of the research that we're going to be talking about today. But then you can also see there's quite a few other people. We have and other organizations, Syngenta, Bayer, BASF, ABA, all provided product and or additional funding, supplemental funding. The soybean checkoff has also been very helpful. And then also we had sort of a shift halfway through our project, and we added a partner, Pattern Ag, to look at some of the DNA patterns in the soil and how that related to SDS. All right, so for those that are not familiar, sudden death syndrome is a disease of soybean, and it's caused by, at least predominantly caused by the pathogen Fusarium virguliforme, and there are other Fusarium species that can cause it, but we're going to, in general, we're going to be referring to FV or Fusarium virguliforme throughout this talk. It is a soil-borne pathogen, and so it has 2 phases to the disease, and you're going to see me sort of talking about root rot and foliar symptoms throughout. And so the root rot phase starts really soon after planting. In fact, you can see the fungus, identify the fungus within days after planting. And then that fungus will sort of set up shop or reside in that root tissue. It actually, the fungus will never move up to the top part of the plant. What it does is it produces a toxin, and that toxin moves up, and that's actually what creates the foliar symptoms. So the root rot phase, you can see that the 2 soybeans you see in the growing behind the glass, it's what we call Fusarium is what we call root nibbler. So the roots are going to be smaller, and for lack of a better word, they're going to be rotted. They're going to be brown and sort of compromised. If you look at the lower stem, you can split that lower stem, and you can see evidence, sort of discoloration within that split stem. Compared to a healthy one. And sometimes you can actually see that blue sporulation. This is a blue pigmented fungus, and you can see that on the outside of the stem late in the season, sort of evidence that this could be an SDS problem in the field. Now, that's not going to show up every time, so it's not a diagnostic tool that can be used, but if you do see it, that gives you some good confidence that SDS is the problem. To give you an idea, we're going to be talking about different levels of root rot, and in general, the numbers we have usually fall between 10 and 30% root rot, and you can sort of get an idea as to what that looks like, where you start to see some of the, not necessarily the taproot, but some of the lateral roots, maybe parts of the taproot are going to be rotted. But if you get into fields that have a really high pressure for SDS, you can see really significant root rot, as you see the 60 through 100, that bottom tier of roots where they're just progressively getting more and more rotten. And so just to give you an idea of what these numbers are going to look like. Okay, so that's the root rot phase. Again, it produces a toxin. That toxin is going to move up to the top. And then you're going to see at the beginning, you're going to see chlorotic spots or yellow spots. And sometimes it can stop there. But then if the toxin continues to move up, you can start to see this in the sort of the characteristic symptoms of SDS, that intervenor chlorosis and necrosis. And then if it continues to develop, you'll see premature defoliation. And as you get more and more foliar symptoms, you'll see more and more yield loss. The premature defoliation, the petioles will remain. And so that is another characteristic that you can be looking for when you're walking out and looking in patches that you suspect to be SDS. So it does cause yield loss, and that's why it sort of has attention of soybean farmers and ag businesses in general. And it can, depending on when SDS shows up and sort of how severe it is, it can cause flower abortion, poor pod set, fewer seeds per pod, and probably the most common one would just be smaller seed size. So it's really affecting that seed fill late in the season, and that's where you're going to see most of your yield loss. Now, there's other components to SDS that I'm just going to hit a couple of the highlights here to sort of frame in that idea of risk later on. And one of them is soybean cyst nematode. And so if you look at soybean cyst nematode and where it's at in the US and compare it to where SDS is at in the US, they really overlap. And there's a reason for that. Soybean cyst nematode, when it is present, it can make SDS appear earlier and be more severe. And so there is some kind of interaction between these two pathogens. And why we bring that up, or why that's sort of an increased concern recently, the main source of resistance for soybean cyst nematode is what we PI 88788, and that's used in 95+% of the varieties that have the resistance genes for soybean cyst nematode. And we're starting to see that that resistance break down or be less effective against some of the populations for soybean cyst nematode. So we're starting to see more and more reproduction during the season, and we think that reproduction can definitely have an influence on increasing risk for SDS. So one of the ways to manage SDS is we say you need to hit it with as many hammers as you can. And the first one, the biggest hammer, should be looking at resistant varieties. And the next slide, I'll sort of dive in a little bit deeper on resistance, but that's where you want to start. And then we've just talked about the soybean cyst nematode. If you can manage your soybean cyst nematode and keep those populations lower, that can reduce the risk. There are several different cultural practices, planting date, planting population, et cetera, that can influence SDS. And so just sort of be mindful of those. We're not going to go into great detail on those. Certainly, if you have questions about that, I can talk for hours about the other cultural practices. But then also maintaining proper pH, soil fertility levels, that's going to help with having a healthy crop that can sort of reduce the risk for SDS. And then there are some seed treatments out there that work at reducing SDS in fields. So a sort of a dorky plant pathology term we're going to be using in several of the slides, so I'm just going to get it out there, is FDX, that's foliar disease index. And that is a combination of disease incidence, or the percentage of plants that are going to have some SDS, and then the severity. So the sort of how sick or how sad the plants are that do have SDS. And that's usually on a scale of 1 to 9. And so you combine those two numbers and you put it to a scale of 1 to 100. So anything that's, or zero to whatever, is going to be less disease. And if you have 100, that means 100% of the plants have some disease. So, and if we did sort of a big meta-analysis of all of our 6 or 7 years of data. And if you look at the FDX value late in the reproductive stages, 1 FDX equals about 0.5% of yield reduction. So to sort of put it into perspective. And if you look at the 3 images on the bottom of this slide, you can see an FDX of 10 to 20 and it's still easy to see SDS, but you're not going to see any defoliation. You're not going to see any necrosis. Then you get that FDX of 25 to 30. You see, again, 100% of the plants are going to have SDS at that point, and then you just start to see some necrosis, and then you get that FDX in the 50 to 60. You'll start to see defoliation at that point. All right, so resistance to SDS, again, management starts with this. And so just to walk you through, in general, most of the resistant varieties are going to be resistant to the toxin or to that foliar symptoms. Some of them might be resistant to the root rot, but you can certainly have resistant varieties out there that have less foliar symptoms, and you dig up the roots and they don't look that good. But on the flip side, you can dig some up and they actually have less root rot if they are resistant. Surprisingly, we've been breeding for resistance to SDS. We still see it on the market every year. There's still some varieties that sort of get through all of the screens and still make it and have levels of susceptibility that we're not really happy with. And we tend to see that with the introduction of traits. And so and a lot of the traits I'm talking about are for different herbicide packages. And typically when that's done is sort of the disease resistance is sort of lagging a couple of years behind the release of those traits. And so especially when we see new traits being released, you'll see an inordinate amount of varieties out there that are susceptible to SDS. All right, so 2013, 10 years ago, we started looking at SDS seed treatments, and these have been done mostly in the Midwest, Illinois, Indiana, Iowa, Michigan, Wisconsin, Ontario. And in all of these trials, we have a resistant and a susceptible line that's identified by industry. And then in some of these fields are naturally inoculated. Some of them are going to be inoculated, or some of them have natural infestation. Some of them are inoculated, and then some are irrigated, some are not. And then we just look at different seed treatments, and we try to take notes on foliar symptoms, root rot, and yield. So this is just to frame in sort of why we need to get to that risk. And so the expectations of an effective seed treatment, in this case, early on, there was only one, that was Illevo. Even with us identifying fields that had high risk or that we knew would get SDS, we saw about a 32% reduction in foliar disease index, FDX, and about a 7.3% increase in yield. And so this is across 30 locations across several years. That sort of gives you an idea, a baseline of as to, again, these seed treatments are not going to eliminate disease, but they can do a good job of reducing the amount of disease that you're going to have in a field. This is a smaller data set that came a couple of years later because we started looking at that root rot phase and the importance of that. And in this particular case, we had about 41%, almost 42% reduction in foliar symptoms using Illevo. And then if you started looking at that root rot, actually the seed treatment was doing a good job of reducing that root rot phase of the disease as well. We had 38% less root rot. And then in this particular case, it's similar, but a little bit higher yield response, 8.8% yield response using Illevo. And this was across 15 locations in the Midwest. So if you take all of that data, what we did was just sort of a quick economic analysis. And this is, I always say, the plant pathology's version of economics. And we just picked one price. So the price of a seed treatment, and it varies depending on where you're going to get it and what it's packaged with. But we picked it $13 an acre. And then for soybeans, we did $10 a bushel. So using those numbers, what we found was when you had high disease or an FDX greater than 10, almost 90% of the time you get your return on investment. The low disease, between 1 and less than 10, you had about 73% of the time. If you had no foliar disease at all, we had about 43% of the time you get a return on investment. And the no foliar disease, I'm just going to point out, like a lot of what we're seeing in some of our trials is that even when we have no foliar disease, you still might have that root rot phase of the disease, which might be one of the reasons why you still have 43% of the time you're getting a return on investment. The other factor might be that some of these products actually work pretty well against soybean cyst nematode, and so we're planting into fields that even if we don't get SDS, you might see some effect from that soybean cyst nematode. So this is leading us into Dr. Marty Chilvers up at Michigan State. He had a former student who looked at trying to identify if you can see FV levels and soybean cyst nematode levels in the field, and if that would at all be correlated to the amount of SDS and or yield that you're seeing later in the season. And I'm not going to dive into this graph. Mitch Roth, who is the lead author on this, and Marty can probably talk for days about this research, but in essence, what they found was that they could find higher levels of FV and SCN in patches or in areas of the field that had more SDS and less yield. And so that led to sort of the beginnings of this project, where we were hoping to take soil samples, and this is sort of the workflow to get to that risk prediction workflow is take soil samples, and then you extract the DNA, you do qPCR, and that would sort of drop it down into that data analysis, which would go over to that risk level. And pictured here is Ryan Hamilton, who is doing some of that work. Now, the second part is to look at all the field trial data. So looking at what are the SCN levels in the field, not necessarily the DNA side of it, but then sort of collecting as much metadata as you can in that field, looking at the soil, nutrient levels, what are the production practices, et cetera, feeding that into that data analysis, and then looking again, helping eventually get to that risk level. And so that is what we're going to focus on for the next several slides. So what conditions could increase or looking at the risk of SDS and how that could be influenced? We did a study in 2020 and 2021 where we had 123 locations, which is why there was a long list of all of the colleagues that were gracious enough to put this trial out there. And it ranged from Arkansas all the way over to Delaware, North Dakota, down to and over to Ontario down to Missouri. So a wide range. You'll see it in the next slide. There's a map. Most of the fields had a susceptible and resistant variety. Some of them just had a susceptible. All the fields had to include 3 seed treatments: a base plus Elevo, and then a base plus Saltro. And Saltro, so early on the slides were just Elevo, but there is a second product out there, Saltro, that is it's essentially equivalent to Elevo as far as effectiveness. And then we tried to, if SDS showed up, we measured foliar symptoms, we tried to measure root rot in most of the fields, and then yield. So the big difference between this and the product evaluation, though, in the product evaluation, we were trying to find the fields that gave us the best chance of getting as much SDS as possible. In this one, we wanted a wide range. We wanted some fields that have to be that worst case scenario, and then some fields that we think have no SDS or even have no SDS or SCNs. We wanted a wide range of scenarios that we're planting into. And then from there, we're collecting FV, the Fusarium virguliforme, and that's being done by Ryan up at Michigan State. SCN counts were done in the spring of both years and just the fall of the first year. Those were done in our plant and insect diagnostic clinic here at Iowa State. Then we took soil analyses and nutrient testing. We sent those to the Midwest labs. And then sort of a halfway through, we shifted and we used pattern ag to look at some of the soil properties, quantifying the amount of DNA from different pathogens or different beneficials. And so we have some pattern ag data, and then we also have weather conditions. All right, so real quickly, where were these locations? Again, in 2020, we had 64 locations, mostly, we had one in Delaware, but most of the rest in here in the Midwest. In 2021, we had 59 locations. Again, sort of very similar, same list of colleagues for the most part. And then to break it down a little bit, because we're going to, next couple slides are going to be, so these are the locations that had FDX greater than 10. So this, again, mostly here tightly in the Midwest. And then these are the fields that had SCN greater than 2,000 eggs per cc. So again, a little bit wider distribution. All right, so in looking at all of the factors, we had 50 some listed out, and then looking at the different weather parameters, this is the list that came from sort of the preliminary analysis as far as things that were going to predict if SDS showed up in that field or not. And so, and from that list, some things are a little bit surprising. Again, we had fairly low levels of SDS the last couple of years, and so I'm hoping to continue this at some level, just to hit a year where we have a lot of SDS. But in these two particular years, soil pH, soil type, and then you can go down the list. I'm not gonna read through all of these, but some of the things on here were pretty predictable, like planting a resistant variety is going to have less disease. Having soybean cyst nematode present is going to increase the amount of disease that showed up on the list. But some of the things on here I can't really explain, like plant population. If you have a higher plant population, we had more SDS. And so there's some things on here that I think are going to trigger next steps or next studies to try to see if those correlations are real or not. All right, so looking at the data, if you look at all 123 locations, and I'm just going to break it down by base plus Ilivo, base plus Sultro. The yield response was about 1.5 to almost 2% greater using the seed treatment. So this is less than that 7 to 8% that you would see in the high SDS fields that we had in our product evaluation. But if you look at the amount of foliar symptoms that were reduced, that is on par. So we had about 36%, 35 to 37% reduction using both seed treatments, which is pretty similar to previous studies. And the root rot was reduced as well, a little bit less than in the previous studies, but still reduced around 12 to 14% using both seed treatments. Now, if you just focus in on the 16 locations that had an FDX greater than 10, you'll see that that yield increase now is back to what we saw previously in those product evaluations, where the yield increase was around 7 to 8.5, depending on the two products, with no statistical differences. The FDX was reduced around 46%. Again, pretty similar to that second study that I showed in the project evaluation. And the root rot was reduced around 20%. So still not as much as it was in that previous study, but just a little bit more than the overall. And then if you look at the locations with greater than 2,000 SCN eggs, there's 39 locations. Yield increase was a little bit higher with Ilivo compared to Sultro. Again, no statistical differences. FDX was reduced 33% in Ilivo and 16% with Sultro. Again, if you're looking at those foliar symptoms, though, it's less. The FDX is less than 10 in these locations, and then the root rot was pretty negligible for both of the products as far as how they reduced the root rot phase with SCN. So if I find that interesting, where we're starting to see differences when SCN is high, it looks like the products are working less well against that root rot phase of the disease. And so that's again sort of clues as to the next steps when we start researching the next things coming out of this study. All right, so the take home, SDS has two phases. I think it's really important to remember that, because I think that farmers really, and ag business in general, ag professionals really focus on that foliar stage of the disease. And it's not surprising. If you have a bad SDS year, it's really shocking to see fields that just become defoliated really quickly. But there are going to be years, and even using resistant varieties, where I think you're going to see some yield loss in that root rot phase of the disease. I think it's really important to consider in fields that have higher risk for SDS, you can still have some root rot going on that could be affecting yield. Management starts with the resistant varieties, and I'll just leave it at that. Effective seed treatments can reduce both root rot and foliar symptoms, But now the caveat would be maybe we need to look a little bit more as you get increased soybean cyst nematode levels. That might be affecting how effective these seed treatments are on that root rot phase of the disease. And so something, again, that's sort of a next step that we want to look at. Yield response and return on investment is greatest when FDX is greater than 10, and that shouldn't be surprising. And identifying fields with greater risk may help select fields and conditions more likely to require an SDS treatment. And ultimately, that's where we want to go. We're probably just a percentage of the way through all of the data analysis from all of everything that's been collected. And so stay tuned on that. And then as we add in the FV levels that we get from the Chilvers lab up at Michigan State, hopefully we can even fine tune it even more and be able to give people an idea as to when they would need an SDSC treatment to manage this disease. With that, just real quickly, if you want more information on sudden death syndrome or really any other field crop disease, go to cropprotectionnetwork.org. There's a lot of information available on that webpage. And with that, thank you for attending, and I'll be happy to answer questions for the last couple of minutes.
Thanks, Daren. So if anyone has questions, please type it into the question and answer session, or I'll be forced just to make up questions for Daren. So I'll try a hard one. So you talk about resistant varieties. I think most of us have heard about like SCN resistant soybean varieties. Are these different ones that are, how's the resistance work?
Yeah, so I mean, in general, it's more difficult to get resistant varieties for SDS, and for a couple of reasons. One, it's an inconsistent disease, and so you could have a really kick butt nursery, and then just like in our research trial, sometimes we don't get disease if the rain doesn't show up at the right time or something, and so you might have years where you just miss data, and so sometimes the landscape becomes the testing ground. Like we'll have general ideas as to how well that it should hold up, but sometimes varieties sneak through. And so it's always good to be, I always tell people the best data is you walking into your fields in August and taking notes on what works and what doesn't work, and going back and talking to your seed dealer and sort of fine tuning what you want to pick the next time. So.
Okay, thank you. I'm giving time for anyone to type in the question. So this would be resistant then to just fungal diseases in general, or are they just kind of plant stuff and see what works out well?
Ask that again.
So are these like resistant to just like fungal diseases, or is this a matter of they're just testing different varieties, but they don't know the mechanism of the resistance?
No, I mean, I think that they established many years ago that resistance is probably to the toxin. And so like and if you think about it, like if you want to screen 10,000 lines, you sure as heck don't want to go out and dig up 10,000 roots from 10,000 plots and then wash them and rate them and figure out which ones are best. There's a lot of really kick butt research being done here at Iowa State in the soynomics group that's looking at root health and trying to modernize how we take notes on root rot. And maybe we can get there, but as of now, it's a whole heck of a lot easier to just wander through a field and look at which ones have foliar symptoms. And so so I would say right now they're resistant to that toxin, and very specifically. So.
Okay, very good. And that we do have a question in the thing that kind of relates to that from Mesfin Azine. Is it known how the fungus benefits from the toxin that affects the leaves? Why does it produce the toxin?
Pass. So there's a lot of work being done by Madan over in agronomy here at Iowa State that is looking at the toxin and the interactions there. I wish I knew why there's certain Fusaria that choose to produce a toxin and then sort of destroy parts of the plant that it's not even going to, or if it just happens to be that's just an unfortunate correlation. I have no idea.
Very good. Another question from Peng Tian. For real-time PCR to detect Fusarium, are there probes specific for SDS?
Yes, and so there is a qPCR technique. We actually published a paper 5 or 6 years ago where we were testing 5 or 6 different qPCR protocols that were published, and so there's a lot of labs that were being done, and the one that ended up being the most consistent and the most accurate was the one out of Michigan State, and so it sort of become the standard, is the one that Marty Chilvers developed and we're continuing to use. So yes.
I'll just ask a follow-up to that before the other question from Peng. Any chance to detect other Fusarium, such as Fusarium graminearum and merid? I'm just gonna mangle the.
I'll save you.
Thank you.
Yeah. So and if you look at the Pattern Ag company, when they're doing theirs, they actually have they have 2 different panels they're looking at. One is Fusaria in general, and then one would be Fusarium species that produce that toxin. So again, it's just not virguliforme. There's a couple of other ones that have been published, and so there are probes out there that can identify, sort of on a broader sense, Fusarium in soil. So
okay, and a question from Melanie Terraher. Did you find that you had other diseases hit your plants after SDS, and if so, were they more severe this year?
At least I'll just speak for Iowa, and in the last couple of years, we've been inordinately dry, and so we did get a little bit of SDS showing up very, very late. We had some August 20th rains that drove some disease, but as a plant pathologist, I was a little bit sad as to how much disease was showing up, which probably meant the farmers were a little bit happy. But yeah, we've had very little disease of almost everything the last couple of years, just because of the dry conditions.
Okay, and it looks like she had noted that in Missouri, plants that were hit were SDS. Later in the season, Cercospora hit hard as well. Will there be any correlation or?
I mean, the only correlation would probably be that both fungi like it rainfall, and so if you have the conditions for SDS, it's high yielding conditions, which means you have enough moisture to sort of invite other fungi into that party.
Great. Another question: if a farmer does not change his management strategy, how often, in terms of years or percentage of years, will severe SDS occur? In other words, how often can I expect to see severe SDS in a field with severe SDS last year or the year before?
So I say, that's a really good question, first of all, and there's not an easy answer. I would say that if you had SDS in your field at any point, that means you have some risk in the future. And I know it just seems like a no-brainer, but it's really hard to get rid of it. And so some people will try to say, well, if I plant 10 years of corn, then maybe I can come back and not get that really bad SDS field. We know that this pathogen can survive and reproduce on corn residue. And so unless you're rotating away to some alfalfa or something that is not corn, I think that you can't really rotate away from the pathogen or reduce that risk. That being said, like I have fields on some farms that were my favorite fields, and then it's been 5 or 6 years where I haven't had good disease in there. So there is some fluctuation as far as other cooties, other beneficial organisms that might be in there that might outcompete with Fusarium virguliforme. And so we have a lot of work that still needs to be done, trying to figure out what all the nuances are in that. And we had a long-term rotation study here in central Iowa that we were collecting. I don't know how much soil is left in the field. We collected so much soil out of there, and we keep testing it, trying to figure out if we can find why some rotations and tillages and stuff like that will reduce Fusarium virguliforme in that soil and others don't. But we have a lot going on.
Great. So another question from Danny Sackett. Do you see SDS in corn, wheat, and other crops to the same extent as in soybeans? And the question's thinking about crop rotation purposes.
So no, so I don't know about wheat, but corn can get infected. So we just published a paper on that. It's not affected by Fusarium virguliforme, but it can be a host, a non-symptomatic host to that pathogen. And we consistently see SDS being greater in fields that were corn the previous year. Not even a soybean rotation can reduce SDS more than a corn soybean rotation. So as far as wheat, I'm not entirely sure. We know that we did a host crop study in the greenhouse, and pretty much everything can. Fusarium is a pretty general pathogen. It can get on almost every crop, but it's not going to really be damaging to them except for soybeans, dry beans, some of those related things to soybeans.
Okay, thank you. I think you've answered all the questions. So thank you so much for the webinar and answering all the very challenging questions that you had. So our next Pests in Progress webinar will be Wednesday, January 18th. There's no webinar in December, again at 12:30 Central Time. Our next webinar will be Saving Money and Pesticides for Apple Growers with New Spray Technology and Warning Systems. So we'll have Dr. Mark Gleason and Liv Meyer from Iowa State University and Melanie Ivy from Ohio State University presenting that one. So thank you all so much for attending. Thank you again, Daren. This was a great webinar, and have a great rest of your afternoon, everyone.